Welcome back to Antibody Basics by Biointron, Part 15. In this episode, we'll talk about therapeutic targets for neurological disorders. Antibody-based therapies are increasingly used to target molecular mechanisms in neurological diseases. These therapies address pathogenic proteins, immune dysregulation, and neuroinflammation. Key focus areas in target identification are antibodies designed to neutralize toxic protein aggregates, such as amyloid beta, tau, and α-Synuclein. Mechanisms of action include direct antagonism, immune modulation, and enhanced clearance of pathological proteins. Some clinical advancements and challenges include FDA-approved antibodies for neurodegenerative and autoimmune disorders, and blood-brain barrier penetration, off-target effects, and efficacy limitations in progressive disease stages as the challenges. The blood-brain barrier is a highly selective barrier that limits the entry of large molecules, including therapeutic antibodies. It maintains the central nervous system's homeostasis, preventing unwanted immune responses and potential toxicity. Key challenges for antibody therapeutics is their size limitations, as antibodies about 150 kilodaltons are too large to cross the blood-brain barrier via passive diffusion. Active efflux transporters like P-glycoprotein also reduce CNS drug accumulation, and less than 0.1% of systemically administered antibodies reach brain parenchyma. Strategies to overcome blood-brain barrier limitations include receptor-mediated transcytosis, which includes engineering antibodies to target transport receptors, or nanocarriers and lipid-based delivery, which involves encapsulation to enhance penetration and stability, or intrathecal and intranasal administration, bypassing systemic circulation for direct central nervous system delivery. Chronic neuroinflammation contributes to neuronal damage and disease progression in conditions like Alzheimer's, Parkinson's, and multiple sclerosis. It is driven by activation of microglia, astrocytes, and pro-inflammatory cytokines. Therapeutic antibody approaches include targeting pro-inflammatory cytokines with anti-TNF-alpha antibodies or anti-IL-6R antibodies, or by depleting pathogenic immune cells with anti-CD20 antibodies or anti-CD52 antibodies, for instance. Challenges and future directions include balancing immune suppression and neuroprotection, thus reducing inflammation without compromising immune defense, or enhancing CNS penetration by improving antibody delivery to inflamed brain regions, or with personalized approaches such as biomarker-driven therapies for optimized patient response. Pathological hallmarks of Alzheimer's disease include amyloid beta plaques, which are extracellular deposits contributing to synaptic dysfunction and neurotoxicity, or tau tangles, which are hyperphosphorylated tau proteins leading to microtubule destabilization and neuronal degeneration. Therapeutic antibody strategies include anti-amyloid beta monoclonal antibodies like aducanumab, which are FDA approved for reducing amyloid beta plaques, or lecanemab and donanemab, which target different amyloid beta conformations to enhance clearance. There are also anti-tau antibodies such as gantenerumab, which aim to neutralize tau aggregation and propagation. Next, we'll move on to Parkinson's disease. Alpha-synuclein aggregation leads to Lewy body formation, synaptic dysfunction, and neuronal death. It spreads in a prion-like manner, propagating pathology across brain regions. It is a major target for immunotherapy due to its role in disease progression. Therapeutic antibody approaches include passive or active immunotherapy. Passive immunotherapy with monoclonal antibodies, such as prasinezumab, targets aggregated alpha-synuclein to slow disease progression. Other examples include cinpanemab, which unfortunately failed to show clinical efficacy in phase 2 trials. Challenges for these monoclonal antibodies include limited brain penetration and potential for immune-related side effects. Meanwhile, vaccination strategies with epitope PD-L1A induces an immune response against α-Synuclein, which is advantageous for potential long-lasting immunity, but has variability in patient immune responses. Multiple sclerosis is a chronic autoimmune disorder leading to demyelination and neurodegeneration. B cells play a key role in driving inflammation and central nervous system damage via antibody production and cytokine secretion. Therapeutic antibody strategies include anti-CD20 monoclonal antibodies for B-cell depletion. For instance, Ocrevus is an FDA-approved antibody for relapsing and primary progressive multiple sclerosis. Kesimpta is a subcutaneous B-cell depleting therapy, and rituximab has an off-label use, which is effective but not officially approved for multiple sclerosis. Other approaches include anti-CD52 antibodies or anti-VLA4 antibodies. There are now several emerging targets and next-generation antibody therapeutics in development. Novel antibody formats include bispecific antibodies, VHH antibodies, and antibody-drug conjugates. Advances in blood-brain barrier penetrant antibodies include advancements in receptor-mediated transcytosis and FcRn inhibitors. Future directions include AI-driven antibody design, combination therapies, and expanding indications. Biointron focuses on antibody discovery, expression, and optimization. From gene sequence to purified antibodies, it takes just 2 weeks. Biointron's high-throughput platform enables the rapid expression of recombinant antibodies in HEK293 or CHO cells. We guarantee the delivery of 100 micrograms to 100 grams of recombinant antibodies. Biointron is also a well-recognized leader in VHH antibody discovery and production. With our self-owned alpaca breeding farm, we offer a 1 alpaca for 1 project commitment and guarantee the delivery of multiple binders, high diversity, and large capacity. Biointron also has several other services, including but not limited to bispecific antibody production, where most formats can be produced, with thousands of bispecific antibodies already successfully delivered. Afucosylated antibody production, as afucosylation has been shown to significantly boost ADCC effects. Single B-cell screening, with 2 to the 10 by the power of 6 plasma B cells from immunized animals, hybridoma sequencing, cross-verified with 5 clones and 100% sequencing accuracy guaranteed, and both antibody humanization and affinity maturation for antibody optimization, as well as recombinant protein expression in the mammalian system, and a high yield guaranteed with our ECA-CC licensed CHOK1 stable cell line. Thank you for watching, and comment with any questions or topics you'd like us to cover next. Don't forget to like and subscribe for more.